and PhD at Yale University before going on to complete his internal medicine residency at Brigham and Women's Hospital, followed by his medical oncology fellowship here at Dana-Farber. At that time, he also completed postdoctoral training in the lab of David Livingston. After that, he went on to Rutgers and Robert Wood Johnson Medical School, where he ran an independent lab and was a practicing breast oncologist. His research includes investigating genetic instability and epigenetic instability with a special focus on BRCA1 and BRCA2. And he also has expertise in translational cancer genomics and biology of rare cancers. He had a lot of leadership positions at Rutgers, but most excitingly, he's just moved on to NYU Perlmutter Cancer Center, where he's now the director of the Center of Molecular Oncology. I don't want to take too much of his time, so without further ado, I'm really excited to announce Dr. Ganesan.
Thanks so much, Sarah. It's a real pleasure to come back to the Farber. I think this may be the second time I've worn a suit jacket at the Farber. And I know back in the days that I was a fellow here, I actually had long hair and a ponytail because, remember, so those are distant days. But it's great fun to see a lot of my old friends, colleagues, and mentees here. And today I'm going to tell you a couple of stories. I can't come here to the Farber without giving a little bit of history of the biology of BRCA1 and BRCA2 and what went on in David's lab. This is the house of David in my view. In any way, as well as some new stuff that came out of the principles that came out of our study of BRCA1 and BRCA2. And I'm going to give you a clinical story and then a little bit more basic story from this. So these are my disclosures. I consult for a variety of things. My wife is an employee of Merck and has equity in Merck. And so I have to pause here. This is David Livingston, who I did a postdoctoral fellowship with. I was incredibly lucky when I was a resident at the Brigham, and Marshall Wolf saw that I was interested in oncology because I saw the incredible clinical work of both Rich Stone and Judy Garber. He said, "Oh, you need to do some science. Go see David Livingston. He knows everyone." All right? And so I met David, and I was a schlep. I had no idea who he was. But somehow within 10 minutes of meeting him and him, writing, maniacally on the whiteboard, I had somehow agreed to work in his lab on a new project on BRCA1, a novel tumor suppressor, and work with Ralph Scully, a postdoc, was working there. And that really changed my science going forward. And this is David in his natural habitat here with Bing Shah, who cloned PALB2, Sharon Cantor, who found BREP1, Bach or Fenk J, and Ronnie Drapkin, who together with Chris Crum found the cell of origin of what was called ovarian cancer. And so a little brief background I'll tell here, breast cancer is common, and a small proportion of breast cancer is associated with familial disposition. And if you look at families that have a high incidence of breast cancer, in those families, again, a small proportion, about 15%, and maybe a little bit more with PALB2, are due to Mendelian high-risk genetic predisposition due to mutations mostly in BRCA and BRCA2, and a little bit of contribution from other genes, and perhaps polygenic factors. But actually, minority of those are due to these Mendelian risk factors in BRCA and BRCA2. And if you look at the human BRCA1 cancer syndrome, as we know, women carrying a mutant allele of BRCA1 have a greatly increased lifetime risk of breast and ovarian cancer. And the tumors that arise have all essentially lost the wild-type copy of BRCA1. So this is functioning as a classical tumor suppressor. All right? And interestingly, men with BRCA1 mutation have little to no increased cancer risk. And if you look at the cancers that arise in mutation carriers, cancers tend to have early age of onset. So this is, the bottom line here is the incidence of breast cancer in the general population. And the top line here is the cumulative incidence of breast cancer with age in a BRCA1 mutation carrier. And the next two lines are BRCA2 and PALB2. And you see these high-risk genes, the increase of incidence really occurs in the 30s and peaks and goes up in the 50s, and then from 50 on, the curves are parallel to the general population. The risk is really in these younger women. All right, and the kind of breast cancer that arise in BRCA mutation carriers are not any breast cancer. They tend to be mostly high-grade tumors. The majority, but not all, have a triple-negative phenotype. And it's interesting, BRCA1 I always consider, is an accidental tumor suppressor in many ways, because you imagine that, if you look at, there's no premalignant. If you look at a prophylactic mastectomy specimen or a prophylactic fallopian tube specimen in someone who's undergone prophylactic surgery, there isn't it's chock full of premalignant lesions. Not necessarily that we understand what the premalignant lesions are. all right, in this disease. And if you imagine a woman who has, all trillion of her cells heterozygous for BRCA1, there's only, interestingly, a 50 to 80% chance that one of those cells will get the acquired changes to become cancer. So this is not P53, where you get cancers in early life. This is not APC, where by the time you're 30, your colon is full of polyps. This is not NF1, where you get multiple premalignal lesions. This is in a per cell level basis, it's actually cancer is relatively rare. In an organism, obviously, it becomes frequent. When BRCA1 was cloned and identified in the early 90s, to everyone's surprise, it coded for this large, 1,800 amino acid polypeptide that had no homology to any other gene that had been discovered. And everyone thought, because of its biology associated with breast and ovarian cancer, that it would be lineage restricted, have something to do with the biology of the breast or the ovary. And to everyone's surprise, it was not lineage restricted, but instead cell cycle restricted. So any cell that passes G1 transition expresses BRCA1 in what Ralph and I saw those days, these little beautiful nuclear foci. So it was not lineage restricted, but cell cycle restricted. And it was an unusual tumor suppressor in that if you acutely take cells and remove BRCA1, they didn't grow better, like with P53. Instead, you had rapid cell cycle arrest that's partly P53 mediated, and you get the generation of gross chromosomal abnormalities. All right? And of course, interacts with many proteins. Interesting, when BRCA2 was then cloned and identified a little later, all right? And remember, BRCA1 and BRCA2 were identified by old-fashioned genomics as loci in different chromosomes, and there were breast cancer 1 and breast cancer 2. When this was cloned, again, this encoded for an even larger polypeptide, over 3,000 amino acids, and it had no homology to BRCA1. All right? And so even though BRCA1 and BRCA2 are 80% homologous by name, they're 0% homologous by sequence. All right? They are different genes that, weirdly enough, had a similar but distinct but somewhat overlapping tumor syndrome. And again, just like BRCA1, it again was not lineage restricted, but cell cycle restricted. And widely, early data from Junji Chen and Dan Silver in David's lab, again, found that BRCA1 and BRCA2 really co-localized, came up in S phase and co-localized in these nuclear foci. And again, like BRCA1, acute loss of BRCA2 led to cell cycle arrest associated with gross chromosomal abnormalities. So when people ask, what potential functions BRCA1 and BRCA2 might participate, there's a variety of different functions that people have identified. But the bottom line is that if you go through a few cell cycles without BRCA1 or BRCA2, your chromosomes fall apart. All right? And so this is a spread of a mouse embryonic fibroblast in which BRCA1 is acutely removed by a Quilox system. And if you remember, mouse chromosomes are acrocentric and normally nicely well paired. Because here, these cells that have gone a few cell cycles without BRCA1, you see now chromatid losses, aberrations, quadraradial, triradial chromosomes. And that's what happens when you go through cell cycles without BRCA1. Or in fact, BRCA2 has a very similar phenotype. All right? And the role for BRCA1 in these processes was actually initially found by some hints. Ralph Scully found that BRCA1 localized in nuclear foci, especially after DNA damage with RAF51. Suggesting, it's probably involved in some sort of homology-mediated repair. And early work by us showed that BRCA1 deficient cells had a profound defect in DNA double-strand break repair, here showed by old pulse field gel electrophoresis. All right? And then work by multiple labs, including the laboratory of Maria Jason and others, showed that BRCA1 was really involved in a particular form of DNA double-strand break repair called homology-mediated repair. If you remind, when you have a DNA break, all right, a DNA double-strand break, the cell has two problems. One is structural integrity. The chromosome is broken in two pieces. You got to put the pieces back together. All right? And the second is informational loss. Often DNA double-strand breaks, there's staggered single-strand breaks, and the ends are processed and cleaved. And so you lose nucleotide information. So in addition to a structural challenge, there is an informational loss. All right? And the cell, for error-free repair, has to solve both the structural problem and the information problem. It can easily solve the structural problem by NHEJ or SSA, where basically you either trim back the ends a little bit or not at all, and then slam the ends together. There are the chromosomes back together. All right? But you've lost the information that's lost in that area. The only way to regain that information is to search in the cell for where that information is still present. And in S phase, the sister chromatid has that information. And if that's present, you can use the sister chromatid to restore the missing information through homology-mediated repair. All right? And this pathway is incredibly important in mammals and higher animals because you can lose NHEJ and SSA, and actually the cell and the organism is fine. All right? but you lose BRCA1 or BRCA2 or F51, you now cannot undergo homology-mediated repair during S phase. You instead shunt towards these error-prone mechanisms, and your chromosomes fall apart. All right? In a manner that we don't quite yet understand, somehow cancers in the setting of P3 mutations are able to survive BRCA1 and BRCA2 loss and struggle through S phase. And to be honest, I still don't really understand how tumor cells survive that, all right? Because normal cells, you take it out, they all rest. And people have shown that the BRCA1 deficient tumor cells are exquisitely sensitive to certain forms of DNA damage, which require intact homology-mediated DNA repair mechanisms. And the first example of this was found by Alan Ashworth and Thomas Halliday to show that PARP inhibitors killed BRCA1 and BRCA2 deficient cells. And if you guys remember, PARP is poly-ADP ribose polymerase. It's a highly abundant chromatin protein, and basically when it senses single strand of DNA, it turns, it creates these poly-ADP ribose chains. All right, it consumes ATP, makes ADP ribose. And that is thought to be a signal for basic excision repair and breaking GIPs. But it's actually turned on anywhere you see single-stranded DNA, including at promoters. All right? And the original story for this was called synthetic lethality, a term I didn't like, in which they thought, what happens is if you have a PARP inhibitor or you lose PARP, spontaneous DNA single-strand NICs are not repaired efficiently. This is no problem. The cell doesn't care about that. But if the cell then goes into S phase, and the single-strand NIC hits a replication fork, you theoretically get a DNA double-strand break equivalent. But that's no problem because the BRCA1 and BRCA2 are intact. You can use homology-mediated mechanisms to restart, and the cell's fine. However, the cell does not have intact BRCA1 and BRCA2. This creates a problem for the cell and can cause catastrophe. That was our first story on why this works. Second story may be more complex. They said, well, actually, the lesion is not the unrepaired single-strand break, but perhaps it's a PARP actually trapped on DNA. And PARP inhibitors the effective ones, trap PARP on DNA, and it's the bulky adduct of PARP-bound PARP that's trapped by DNA that's now the bulky adduct. And that's the lesion that BRCA1 and BRCA2 repairs. And that's our second story on how this worked. We still don't quite understand this because now there's, I won't go into the details, but now there's data, perhaps replication gaps or perhaps transcription replication fork interference may be the lesion of how PARP inhibitors work. All right, but clearly they do in this setting. All right? And intriguingly, this is one of the few times where a Nature paper that was done in 2005 was converted into a phase 1 study that had a New England Journal of Medicine paper in 2009, so 5 years from Nature paper to New England Journal of Medicine paper. All right, and so this is wild because this is one of the first phase 1 trials that's published in the New England Journal of Medicine. The entry was not a tumor type, but a mutation. So you just had to have a germline mutation in BRCA1 and BRCA2, and they included cancer variety of histologies, and they saw responses in these advanced cancers. All right, and then this led, after a little bit, if you guys know the history, there was a little bit of a sidetrack into the entire field was sidelined by the development of iniparib, which was a PARP inhibitor that wasn't. All right, it was a drug that actually was not a PARP inhibitor. All right, and that sidelined the field for a while. But ultimately, after we recovered from that misadventure, ultimately then PARP inhibitors came into practice, and now they're approved in a variety of settings in ovarian cancer, breast cancer, pancreas, and prostate for germline BRCA and BRCA2 genes. Unfortunately, and interestingly, the potency of PARP maybe increases with PARP trapping, which is intriguing. So talazoparib is one of our most potent inhibitors, whereas veliparib, barely traps and barely has any efficacy either in cell lines or in vivo. And that also told us that other chemotherapy agents that induce bulky lesions are also highly effective in BRCA and BRCA mutant cancers. So platinum, but also topoisomerase inhibitors, old bifunctional alkylators. Actually, the best thing that works best in vitro is actually things like BCNU and CCNU in BRCA mutant cells, all right? And even anthracyclines. So PARP inhibitors aren't magic. These other drugs also work well in these diseases, and we'll talk about this, all right? But despite the development of these inhibitors, all right, ooh, cool. I think that's the theremin, right? Noise. All right. It's an old. But despite the development of these, BRCA1 and BRCA mutant cancers can actually develop resistance both to platinum and to PARP inhibitors and other DNA damaging agents. And the mechanism of acquired resistance is actually incredibly informative and tells us a lot both about the biology of these genes and their role in tumorigenesis. And so if you take BRCA2 mutant cells, bathe them in platinum, ultimately you'll get cells that are completely resistant. Similarly, if you take BRCA1 mutant mouse models, this is with Jos Jonkers in our lab, and you treat them with olaparib, you can get this beautiful response, but ultimately you get resistance. And the mechanism of resistance are really informative and frightening. So the first mechanism of resistance is reversion mutations. Okay? And so reversion mutations, if you remember, most pathogenic mutations in BRCA and BRCA2 are small indels which cause a frame shift. And the frame shift theoretically takes the gene out of frame and you get a premature stop codon, which theoretically can create a truncated protein. But often, actually, you don't create a truncated protein. These are functionally null because of nonsense-mediated repair. If you guys remember, if you have a stop codon that's within a certain distance of a splice acceptor region, that is tagged as a bad RNA, and the RNA is degraded. So unless the frame shift is in a large exon or in the terminal exon, often nonsense-mediated decay will degrade the RNA, and you get a functional null. Right? So if you take a cell that has this kind of truncating mutation in BRCA2, this is work done by Alan Ashworth, but also simultaneously done by Toshi Taniguchi, and bathe them in platinum, ultimately you get cells that are resistant. If you take the resistant cells and resequence BRCA2, what we found is the resistant cells have acquired a secondary mutation in BRCA2 in which the exon containing the frame shift has been excised, and the rest of the gene is placed back in frame. And so this converts a functionally null frame shift into an internal deletion and creates a hypomorphic protein that is functional in repair. And so this is wild, a second mutation that restores function. All right, and this shows, this is in cell lines in BRCA2, and I'll show you this is work by the Swisher lab that showed this in humans with BRCA1. So this is a ovarian cancer that at diagnosis had 2594 del C, so a single nucleotide deletion, so it takes the gene out of frame. All right, and this was sensitive to platinum. When the tumor acquired platinum resistance, it was resequenced. And now, in addition to the single nucleotide mutation, deletion, there's now 23 extra nucleotides that were deleted downstream. All right, so 1 plus 23 is 24. 24 is divisible by 3. The gene's back in frame. All right, wild. All right, and so this converts a functionally null frame shift into an internal deletion of 8 amino acids plus some new amino acids in there as well. And if you look at the edges of these deletions, there's microhomology. So what's going on is the deletions are generated by inappropriate NHEJ. That's the mechanism that's inappropriately used in these BRCA and BRCA2 deficient. So these deletions that these BRCA1 2 mutant cells are prone to developing are in fact the mechanism of the secondary mutations that cause reversion. The other mechanism of resistance is compensating mutations. This is work done simultaneously by Sam Bunting and Andre Nuzenzweig's lab, as well as my laboratory and the laboratory of Jos Jonkers and Madeleine Tarsunas. And we won't go into details, but this is kind of wild function where through various screens we found that mutations in an NHEJ protein, 50BP1, can actually restore HR function in BRCA1 mutant cells. All right, it's kind of wild. How can a second mutation in another DNA repair gene restore function? How do two wrongs make a right in this setting? And actually, without going into the details, I'll show the mechanism that was ultimately figured out that showed that BRCA1 was really a regulator of repair choice. And the way to think about that is in order to figure out how to repair a DNA double-strand break, it all depends on how the ends are processed. The ends of the breaks are processed. If you can't process the ends, the only thing you can do is slam them together. And 53BP1, which is expressed during all phases of the cell cycle, we think now one of its roles is really to prevent end processing, to prevent the CTIP and mRNA complex from chewing back the ends and allowing revealing single-strand DNA. And if you do that, all you can do is slam the ends together. During S phase, now BRCA1 is expressed. And one of its functions of BRCA1 is inhibiting 53BP1's function in suppressing end resection, and so allows end resection to take place. And now you have end resection, now you have an end free for homology search, and you can initiate HR. If you lose BRCA1, then 50BP1 is free to do in S phase what it does outside of S phase, which is suppress end resection. And so again, you can't resect the ends. All you can do is slam them together. You get NHEJ and the genomic chaos that ensues. But if now if you lose 50BP1 as well as BRCA1, now the CTIP and mRNA complex can be turned on by phosphorylation during S phase, and it's allowed. You actually get hyperresection, but that's tolerable, and you can restore HR. All right, so it's kind of wild. It's a compensating mutation that actually restores HR, all right, by doing this. And it shows that BRCA1 is not required for the nitty-gritty of homology-mediated recombination, but really for kind of a repair choice that's being made, all right, is one way. BRCA2, you can't rescue it this way because it plays a role way, all right? And so if you think about this, there's several mechanisms required resistance to PARP inhibitors and things in BRCA mutant cancers. There's drug-specific mechanisms. Okay, so for example, olaparib is a beautiful PGP substrate, so you can just upregulate that, or you can have mutations within PARP itself. All right, and then there's pathway reversions, either by direct genomic reversion or compensating mutations. And it's important to understand the mechanism of required resistance, because if drug-specific, you can just change the drug. But if it's pathway reversion, it becomes more difficult, all right, on how to do this. If you have true genomic reversion, you may not necessarily have a handle. And there are ways like POL, theta, things, others that are going there, which people are working on. But the more important thing is, what do these reversions really tell us about the biology of BRCA1 and its role in tumorigenesis? One thing it tells us is that, hey, in addition to PARP inhibitors, platinum, PARP, anthracyclines, topoisomerase inhibitors, they're all targeted treatments because all of them induce reversion mutations with selection, all right? And that's the genetic definition of targeted therapy, right? Under selection, you get a second mutation that restores function, all right? So these are targeted therapeutics, all right? And if we think about that, as targeted therapeutics, maybe when chemotherapy in general works, it's not a gross metabolic poison, but is in fact a form of target therapy for an underlying defect in DNA repair checkpoint control. All right, we'll talk about that. The other thing is that BRCA1 mutant cancers can totally reconstitute BRCA1. So we call this tumor suppressor tolerance. You're not going to cure a BRCA1 mutant cancer by putting back in BRCA1. All right, it's going to be fine. This is not p53. And the third thing is that, this also, which is a corollary of the second one, is that DNA repair defects are only required for initiation. So loss of BRCA1 is required for initiation of tumorigenesis, but not required for maintenance, because you can revert, and the tumor is fine. Right? In fact, it's happier. All right? And that tells us, so that raises another question, what's driving the growth of these BRCA1 mutant cancers? All right? And so I'll talk about these questions that these problems come in, and how we try to address that. One is that, hey, maybe DNA-damaging chemotherapy is targeted therapy aimed at underlying defects in DNA repair. If so, the way we dose chemotherapy is all wrong, right? We go to maximum tolerated dose, and we say chemotherapy everyone hates because they say it makes people sick. And the important we realize is that chemotherapy is toxic because we made it toxic on purpose by going to maximum tolerated dose. There's no other class of medicines that we use that we go to MTD. We don't give you the MTD of Tylenol. We don't say, listen, necrose your liver and back off a little bit. That's the right dose. All right? All right? And why do we do this? It's based on mathematical modeling of Burkitt's. All right? Basically, the phenomenon was that, hey, these are gross metabolic poisons, toxicity is on target, and we're going to use toxicity as a pharmacodynamic marker of efficacy. Literally, the more bitter the pill is, the better it is. So as bitter as you can tolerate is the best dose. All right? And what about dose density is basically based on mathematical modeling of Burkitt's lymphoma, where it's clear that in Burkitt's, it was cured by high dose and dose intense treatment regimens. And every time we backed off on either dose or schedule, cure went down. And the reason for that was this great mathematical modeling saying that, hey, these are in log phase growth and you need to have multiple things to take it down below an effective threshold to affect cures. And this kind of approach is the reason why we do this kind of chemotherapy in all of solid tumors. All right? But the funny part is that the poster child for which this is based on, we actually do things differently now. All right? And so when I saw this paper from Dunleavy et al. In 2013, which was the modified EPOC and the dose-adjusted EPOC dose, this low-intensity therapy for adults with Burkitt's. And so what they decided was that, hey, Burkitt's therapy is great. It cures children. But if you give it to the elderly or patients with HIV, they die from the treatment. All right? So that's obviously not what they want to do. So they said, okay, let's do this. And so one of this group did was they said, let's lower the dose that's used in Burkitt's, but instead give it by continuous infusion over 96 hours. All right? And then they modeled the PK. And they showed that, hey, if you give doxorubicin as a bolus, you get this enormous peak, and which is rapidly removed by hepatic clearance, and then a second phase of clearance. This is a standard, half-hour dose, bolus dose. If you instead give it by 96-hour continuous infusion, you avoid the peak, and you get this kind of continuous exposure over 96 hours, and then it falls off. All right? When they did this, this is the freedom from progression. You got, nearly 100% cure. And this really interesting thing, they said, hey, this suggests, hey, it's the length of exposure time above an effective threshold concentration, not the peak concentration is the important therapeutic principle, and that this pharmacokinetic principle may explain why standard regimens in Burkitt's require dose-intense treatment to maximize the outcome. All right, so I said, hey, that makes sense. If there's a minimum effective concentration and you give a chemobolus and it goes like this and that, in order to maintain this, the only thing you can do is give as much as you can repeatedly. All right? Or you can do this and avoid the peaks and do this. So I said, if this is true, can we do this for other tumors that should be DNA damage sensitive, like, for example, triple negative breast cancer? And there are ways, continuous infusions is hard to do clinically, but there are liposomal formulations which can basically cause slow release of doxorubicin. So this is doxorubicin curves, free doxorubicin with liposomal doxorubicin. And so you get, and this actually continues for over a week, you get low levels at about 10 to 100 nanogram per ml, which is what you see with this continuous, which is what you see with the continuous infusion release for days. And similarly, carboplatin versus cisplatin, both this is getting into chemotherapy pharmacokinetics, but it's something I'm interested in as well, is that they both generate the same active compound. It's just that cis generates it in hours, whereas the half-life from carbo to the active agent is about 12 hours. And so when you give a bolus, it gets a slow on and off. All right, and it's the same drug, but all of a sudden carbo, you don't get nausea, vomiting, acute renal problems. All right, so we said, hey, doing this and getting the principle that chemotherapy is targeted agents, can we develop a regimen of carboplatin, liposomal doxorubicin, and triple negative breast cancer that's as effective as a standard anthracycline-containing regimen, typo, more tolerable, and when it works, can we figure out why? All right, look at underlying markers of homology-mediated DNA repair. And so what we did is we did this, and this is internally funded because no one does chemotherapy regimens anymore. So we funded this by ourselves, and we got philanthropy money at Rutgers to do this. And this is done by Deb Topmeyer, Nancy Chan, Kim Hirschfield, and Coraline Mene with a medical oncologist who really did this. And what we did is we took biopsy-proven triple-negative breast cancer. We got upfront research biopsies, then we treated them with just 4 cycles of liposomal doxorubicin and carboplatin at modest doses. We know these doses were effective in metastatic disease. We'd already done a phase 2 trial in metastatic disease. All right? And then we, after just 4 cycles, which is kind of half the standard therapy, we then had definitive breast surgery, and then everyone got Taxol out back because we have to give them Taxol as part of the standard regimen. All right? And then we said, okay, if we do this, how often do we get path-CR? How do they tolerate this? And what are the genomic markers that can predict response? And so these are demographics. We got 54 patients at chemistry. We did this at Rutgers, so we had a very diverse patient population. It's mostly stage 2 and 3 patients. And when we did this, to our surprise, we had a pathologic complete response rate with half the chemotherapy of about 33%, and another had RCB1, which is microscopic residual disease, PREF. All right, and this is actually relatively similar to what you get with a full ACT regimen, which have PC rates of about 20 to 35%. So this by itself, by giving half the chemotherapy in this modulated fashion, is about as effective as standard therapy. And this is the secondary, this is 2-year relapse-free survival was 86%, which is not that far off from the standard full therapy in Keynote 25, the chemotherapy arm, where their arm was 81% versus 87% when you had Pembro here. This is just half the chemotherapy. And this is the relapse-free survival by RCBs. The ones who got PCR RCB 1 to 0 so far, none of them are relapsed. So those have done very well. And this is the important thing. These women did not look like they're getting chemotherapy. So there was, there was little to no alopecia. The only thing they got was grade 1, fatigue, a little bit of anemia. They did get grade 3 or 4 neutropenia, about a third, but we didn't give GCSF up front. So we only gave GCSF if patients became neutropenic. So many of the patients didn't even require GCSF, and we only had 1 patient with febrile neutropenia during this. All right, so incredibly well-tolerated regimen. And do this. So we said, hey, just by using the same drugs, but just modeling the PK and treating it like a targeted drug, we can make chemotherapy less toxic and maintain efficacy with this. And then the second thing is, okay, we wanted to get the residual disease in response with just purely anthracyclines and platinum, because taxane, at least I don't know how that works. even though it works afterwards. So we said, hey, can we figure out then, are there genomic features that would tell you who's responding to these drugs? So we did whole genome sequencing and RNA sequencing of all the pretreatment samples and some of the post-treatment samples, and we looked at the rearrangement patterns. So as you remember, Serena and Xanel showed that if you look at the rearrangement patterns in the genome that are induced in tumors that have BRCA1 loss versus BRCA2 loss, you can see in these sporadic tumors, this is a Circos plot, so chromosomes are on the outside. These little lines are translocations or rearrangements that go from one chromosome to another, or within or internally. I can see this is a BRCA1 wild type, and you see BRCA1s and 2s are full of these rearrangements, and the rearrangement flavors are different. If you look at BRCA1, they tend to have a signature called associated with tandem duplications, whereas the BRCA2 nulls tend to have these deletions of a certain size with microhomology at the ends. All right? And so these rearrangement patterns can actually be quantified, and Serena has made this thing called an HR detect score, which combines the rearrangement patterns together with a single nucleotide parent to understand markers of tumors that tend to be BRCA1 or 2 like. And so if you look in this set of breast cancers, and the BRCA1, 2 mutants are shown in these colored marks, and the HR detect score is shown on the y-axis here, you can see all the BRCA1 2 mutants have this extremely high HR detect score, which means they have a rearrangement signature that's associated with an underlying defect in DNA repair. And then there's a bunch of these that are in wild type that are kind of not quite there as big as the BRCA1, BRCA2 mutants, and there's a bunch that don't have anything, including the tumors that are just heterozygous for BRCA1 and BRCA2. And when we said, okay, in our group, what's there? We can see that in the ones that are RCB 0 or 1 who did very well, the HR detect scores are all elevated, are more elevated, I should say, than the ones that are on the RCB 2 or 3, even though there are some that are elevated here and some that are low here, which we'll talk about. All right? And this suggested, hey, this marker, which is done on formalin fixed exceptions up front, may be able to identify who just responds to this very gentle chemotherapy regimen. And if we look further into detail on what are the exact features of signature that are associated with response, intriguingly, it was deletions at microhomology and the structural variant 5, which is, again, deletions of a certain size. And these are BRCA2-like signatures. And I should point out, in this group of patients that we had, they were all mostly BRCA1, BRCA2 wild type, because at that time we had a different protocol for BRCA1, BRCA2 mutants. So these are all enriched in BRCA1, BRCA2 wild type triple negatives. But even in that, we could see the signature that was really BRCA2-like, not BRCA1-like, that was associated with response in these mostly sporadic wild-type triple-negative breast cancers. Okay, and so right now we are also establishing PDX and xenografts with all the residual disease and looking at the genomic landscape of these primary chemo-resistant tumors. Right? And this tells us something, important. And then this also raises several puzzling questions. In the patients who had path CR, there are a bunch of patients that had very low HR detect scores. All right, and the question is why? This could be a failure of the assay. They truly have an underlying defect in repair, and we've missed it. Or it could be, hey, maybe a defect in homology-mediated repair is one way to be sensitive to these drugs, but not the only way. There could be another repair defect or mechanism that can make you sensitive to anthracyclines and platinums. And can we figure that? So we're using now, in a larger sample set, using discovery to say what makes these samples different from these ones that had no response. And the second is, what's going on here with these guys that have elevated HR detect scores but had RCB2 or 3? Intriguingly, none of these have relapsed so far. All right? So it could be that, hey, one of the things is that maybe these are there and we just didn't give enough chemotherapy. You imagine that if we gave to create a pathological response but still affects the micrometastatic disease. Because you imagine that if we gave 1 cycle, the path CR rate would be much less. If we gave 2 cycles more, we'd get path CR. We picked 4 as a random number. All right, why we came to 4, I don't know. We did, how we came our regimens. And imagine maybe, and for all we know, PK, maybe some of these, we don't look at PK. Maybe these are ones where, with the chronic exposure, maybe these guys are dipping below the threshold that's appropriate. All right, and so these might have been sensitive. Maybe this is due to just dosing. All right, even though interestingly, these ones have elevated so far have not relapsed, even though they have residual disease. All right, and so these are things that we can figure out, to look at what's going on. So conclusion is like, hey, with this, if we think about the lesson from BRCA1, BRCA2, that, hey, sometimes our chemotherapy works when it works, it may be due to underlying defect. We treat it as targeted therapy. we may be able to develop effective, less toxic regimens using rational PK approaches with the understanding that this is target therapy. And more importantly, can we start to develop biomarkers for efficacy of our candidate chemotherapy drugs? All right? When it works really well. All right? But we can't have a 30% response rate. We have to identify those who are going to respond, and then those who respond, modulate it so our therapy is better. All right? And of course, we can complement with functional approaches. We'll need Tony's Latai's help for that. Right. The second story I'll tell you is a little more basic, and we'll go, what do reversion mutations tell us about the biology of BRCA1? Really is that, hey, DNA repair defects are not required for initiation, are required only for initiation but not maintenance. So the big question is, what's causing the growth of BRCA1 and BRCA2 mutant cancers? It's not BRCA1 and BRCA2 because we can revert it, right, and they're fine. And if you look at the genomic landscape, the mechanism of mutagenesis is all these rearrangements. So our one hypothesis is that, hey, is the driver in these tumors, embedded in these rearrangements? So loss of BRCA1 causes rearrangements, and then there's selection for some drivers that are there. But when we talk about rearrangements, most people talk about intrachromosomal rearrangements from one chromosome to another. And you can see, even though these exist in BRCA1 and BRCA2, the majority are actually intrachromosomal, these little lines that go from one chromosome back together. So internal deletions, inversions, transversions are much more common than the kind of things we talk about, which are intrachromosomal translocations. All right? And we often think about in-frame, why are rearrangements important? We always think about gene fusions, right? We think in-frame gene fusions is the way genomic rearrangements cause driver oncogenes. And we know in BRCA and BRCA mutants, they're not present. We don't see many in-frame fusions. So we asked the question, how do we characterize the rearrangements that are present in BRCA and BRCA, in these rearranged cancers? How do we identify genomic rearrangements in cancer? How do we characterize them? For that, I'll tell you, how does next-gen sequencing, which we use for genomic characterizing cancer specimens, how do they identify oncogenic translocations? You imagine you have, for example, here of ALK, and it's rearranged, the N-terminal is rearranged to a partner. How do we figure out the presence that this fusion exists in a tumor sample when we're doing DNA sequencing? All right? And imagine if you do exon capture, it won't make any difference because all the exons are intact. All right? The way certain companies like Foundation and others identify this is that they use intron capture. They use high-depth capture of the introns in front of the kinase domain. And this way, by that, you can actually then capture a junction, all right, that's present in the intron. Now, introns are tough because introns are large. They can be filled with repetitive elements, all right, so they can be hard to work with. But if you design the capture probe enough, you can get 500 to 1,000x sequencing of the intron and then capture the junctions that are found. The junctions are really interesting because by looking at the junction, you can then impute the rearrangement and then see if there's an in-frame fusion, but you capture all the junctions. And the junction also has what happened, how did that break occur, because you have the actual junction sequence. All right. You can use other ways, targeted enriched RNA-seq, SPEC, anchored multiplex PCR on RNA can also pick this up. But since RNA is not well preserved, they're good if they're positive, but lousy if they're negative. All right, they have very good negative control. So at Rutgers, we started sequencing everyone with hybrid capture-based sequencing, actually through a collaboration initially with Foundation Medicine and then ours. And we had a lot of data. And the key thing was we got all the primary sequencing data. And one of the things we looked for was what's going on in the introns that were captured in the tumors that are heavily rearranged that are quote-unquote driverless. All right? And what we found was there was a whole bunch of out-of-frame rearrangements that occurred in these introns in front of the kinase domains that were going off to intragenic space. And they were discarded because it's not associated with an in-frame event. All right? But we were surprised by this because why are they present? Why are they recurrent? All right, and is there a biology associated with them because they're sitting there in well-known oncogenes? All right, and so I'll give you a case. This is PMI 548. So as a woman with metastatic recurrent gastric cancer, was treated with the usual things and successive chemotherapy and had progressive disease. Was sent to our tumor sequencing protocol, and this is the sequencing data from Foundation Medicine, and it showed that it had FGFR2 amplification, but also FGFR2 truncation was how it was reported. We pulled the primary sequencing data, and we found what was going on there was partial amplification of FGFR2, in which there was a rearrangement in which intron 17 of FGFR2 was rearranged to an intragenic area, all right? And so it was just hanging out in space, in intragenic space, all right? And so we said, and this is not the first time we saw this. We saw this multiple times in other cancers. All right? And so, and it was intriguing because FGFR2, unlike many of our, many kinases, when you have gene fusions, often you have the N-terminal fusions where the N-terminal is replaced. Here in FGFR2, all the pathogenic kinase fusions occur C-terminal of the kinase domain and involve intron 17. So always replaces the last exon with a partner fusion. All right? And here was something that was rearranged here, but going off to intergenic space. All right? So no partner. All right? And so we said, we see this multiple times. Is there an RNA that's being generated from this event? And if so, what is there? Okay, and so we did hybrid capture enriched RNA sequencing. And what we found was, so this is a sashimi plot, was that this outer frame rearrangements, actually, the tumors that had it had high expression of an FGFR2 transcript that was truncated, all right, but still present. And so this is a sashimi plot, and what this is these are the individual exons here. This is zoomed up for exon 16 to 18 of FGFR2. And these are the reads that pile up on each exon. And these little bridges that go from one exon to the other are bridge reads. All right, you can see that you're going from 16 to 17, but from 17, you're not going to canonical 18. You're instead having a lot of reads that end up in this cryptic splice acceptor site, which is called the C3 variant. And the genomic break is actually right after it. And so there is no exon 18 for it to splice into in the tumor, so there's still normal tissue here. And so it's splicing into this cryptic acceptor end where there's one amino acid and then a stopper, and then a UTR here. All right? And we're like, that's interesting. So this is generating a transcript, high level of this truncated transcript. Of course, we're not the first person to see this. We went back into the literature, and there was actually a publication back from 2008, in which they noticed, there are these 3 different isoforms that can see in cancer. And the C3 variant, which is the one that we're seeing, was in fact, compared to wild-type FGFR2, or C1, this was oncogenic. All right, and we said, these truncated outer frame rearrangements can create a transcript that's in fact oncogenic. So is this truncated FGFR2 oncogenic? Is it sensitive FGFR inhibitors? And how often are these non-canonical alterations present that are potentially actionable? All right. And so, interestingly, this is one of these cases of serendipity. I always like to tell a story. So we generated these data, and I was actually, this was right before the pandemic. I was invited to NKI by my old friend and collaborator, Jas Jonkers, to give a talk on DNA repair. And he invited me to his lab meeting, all right, to see one of his postdocs present. His postdoc was presenting, this is Jas Jonkers, who's this big animal modeler, and so he's presenting he had made a mouse model of lobular breast cancer in which he got rid of cadherin-1. And he found no phenotype. So he said, there must be a cooperating oncogene. So being Jas Jonkers, he did an in vitro Sleeping Beauty transposon screen to find what events can cooperate with cadherin loss to initiate tumor. And the idea is it's a Sleeping Beauty transposon. It has a splice acceptor, a polyadenylation site, so it lands on an intron, it will capture. It also has potent promoting, so if it lands in front of a gene, it can turn things on. And he turned this on in vivo in the duct cells of a mouse, and then found tumors that arose, and then sequenced to see where they are. And his big hit, which was puzzling to him, was insertions of the sleeping transposon into intron 17 of FGFR2. And he was puzzled. There's a truncation of an oncogene. And when I saw the data, I'm like, we see this in humans. All right, and it's oncogenic. And in fact, so this work done by Daniel Zhang's and Brookins. And so when we did the RNA-seq on those tumors that arose, of course, there was nice truncated RNA. This is 16, 17, and now the RNA is ending in the splice acceptor of the retrotransposon and terminating there. So you're getting a truncated transcript now ending in this artificial retrotransposon. And we're able to show that these truncated FGFR2 transcripts are, in fact, transforming and oncogenic. So if you take wild-type FGFR2 in the top lines here versus truncated FGFR2, either in the setting of, and it had nothing to do with cadherin, either in the setting of cadherin floxed or cadherin wild-type, both of them was potently oncogenic. So truncating FGFR2 is an oncogene. Wild-type FGFR2 is not an oncogene. All right? It's a proto-oncogene. All right? And in fact, the tumors that arise are very sensitive to, you can prolong their lifespan by putting in FGFR2 inhibitors. All right? And the next question is, how often are these non-canonical rearrangements present in human cancer? All right? Because right now they are seen in sequencing, but not even reported. All right? And so this is, on this pie chart here, this is all the FGFR2 alterations that are seen in a foundation data set of about 200,000 patients. And in-frame fusions are seen, if you look at all the FGFR2, There are about present 42 of these. There's like about 1,000 of these alterations. But as many in-frame fusions as there are out-of-frame rearrangements of the intergenic regions, all these kind of things that should create a truncated product that are as many as the ones that are reported. And on top of that, there are these amplifications that are, a subset of them are probably partly amplified and me generating these transcripts as well. And so the in-frame fusion is just one method to create a bad oncogene, but not the only method, and in fact may not be the most dominant method. And if you look in different cancers, how these rearrangements are seen, you can see, it's present in 8 to 10% of cholangiocarcinoma, right, which we know that's where we see these FGFR fusions. But it's also present in 2% of breast, 2% of lung. We think that's a small number, but 2% of breast is more cancers than 10% of cholangiocarcinoma. All right, so by absolute numbers, they're actually dominant in these other cancers. All right, by absolute numbers of cancers. All right, that are present. Next thing we said, okay, these are present and they're not being looked at. So can we, why is this truncated transcript transforming? All right, and interestingly, it's basically you need loss of exon 18 as an auto-inhibitory region, which we found there's actually structural data for this now, which I won't go into. But if you take full-length FGFR2, it doesn't transform, either in vitro or in vivo. If you just make this truncated transcript where you get rid of exon 18, that transforms both in vivo and in vitro. If you put in the classical FGFR2 BIC1 fusion, in which you lose exon 18, so you take that and put it onto BIC1, that transforms in vitro and in vivo. But now if you take full-length FGFR2 and put it onto the fusion partner, so people often say the fusion partner causes dimerization, that's why it causes. But if you retain exon 18 and put the same fusion partner, it no longer transforms and is either in vitro or in vivo. So you need to create an FGFR2 that's stable, that loses this autoregulatory region, either in the setting of a fusion or by truncating the transcript. And to see if this is, once we went to a large PDX data set that was available and pulled PDXs for which we did RNA sequencing and identified tumors that expressed either FGFR2, this exon 18C3 usage, either in the setting of a rearrangement, or sometimes they're just expressed and we don't understand why they're expressed. It isn't clear that there's a rearrangement or duplication that's there. In either of these settings, in gastric cancer, in breast cancer, in liver cancer, when there was expression of this truncated variant, all right, or an in-frame fusion, there was nice sensitivity to FGFR2 inhibitors in this setting, showing that these truncated in truly human cell models, the presence of these out-of-frame rearrangements cause sensitivity. And then the final thing we did was we looked at, we went back to the pemigatinib trial. This is the trial in which the landmark trial, which caused the FDA to approve FGFR2 inhibitor for FGFR2 rearranged cholangiocarcinoma. All right, and interestingly, the criteria was fusion or rearrangement. This is because a long time ago we worked for our foundation, and we told them they have to report all these out-of-frame rearrangements on the first page of their report. So the entry criteria became fusion or rearrangement. All right, and so what we did was said, hey, if we look at the tumors that have non-canonical FGFR2 fusions that have either rearrangement with frame unknown or rearrangement to intergenic space, all right, are they the non-responders or are they amongst the responders? And we found that these out-of-frame rearrangements respond as well when we look back at the tumor set as the canonical in-frame fusions in the tumors that were treated with FGFR2 in this clinical trial. All right, this is a nice collaboration with Insight to reanalyze those clinical data. And we showed that, hey, in humans too, if you have these out-of-frame rearrangements, they actually respond to this. So in addition to the canonical fusion, nonsense or non-canonical FGFR2 alteration, adding those may increase the potential benefits of FGFR2 by 30 to 40%. It's a conservative estimate. We can probably double the number if they're actually reporting these. All right? But this is just FGFR2. We started with it. What about FGFR1, FGFR3, other tyrosine kinases? And we won't go into this. The long story is that we're seeing this true for all the other tyrosine kinases as well. They're out-of-frame rearrangements. They're actually creating stable RNA and protein products that are oncogenic. All right? And to give an idea of this, we went to the RPPA data from I-SPY2. So this is a neoadjuvant deal for triple-negative breast cancers. And we went to Petricoin and Lioto had actually done reverse protein phase analysis looking at phosphoproteins. We said, hey, let's pull the tyrosine kinases, okay, FGFR2. And you can see the vast majority is 200, it's about almost 300 cases. Vast majority have little to no, but there's an outlier, about 6% that have outstanding expression of phospho-FGFR2. And we've shown the truncated transcript, actually, none of these have reported FGFR2 fusion. All right, so we're going to go back, we're pulling these samples and seeing what's going on, what are they expressing? All right, and initial data says that these are the ones that are expressing a truncated transcript, that are not being, they're totally being lost. And we have the same thing, if you look at ROS1, ALK, they're outliers. We're pulling, we're seeing that there's the non-canonical outer frame rearrangements that are creating a stable kinase protein. So, and we're now going back to the BRCA and BRCA2 and saying, are these the missing drivers? These outer frame rearrangements that are hidden within the enormous rearrangements landscape that's induced by loss of BRCA1 or BRCA2, all right, in these tumors. And so, in fact, we started to do long-read genomic mapping and sequencing in the resistant tumors and the primary tumors to look for these in these tumors. And we need this because we know, right now for HR-deficient tumors like BRCA and BRCA tumors, we give chemotherapy, PARP inhibitor, but ultimately we get resistance, right? And we're trying to see, if it's reversion resistance, can we do other things like POLQ, topoisomerase inhibitors, immunotherapy that are non-cross resistant? The other thing we can do is, can we target the actual drivers in the tumor? All right, if this thing is FGFR2, you want to hit with PARP inhibitor plus that to put non-cross resistant selection pressure on the tumor. All right, you need to hit, and these tumors have drivers, they're not magic. All right, just the mechanism of mutagenesis is rearrangements, not point mutations. All right, and right now we're really lousy at identifying that. And so with that, I'll stop. This has kind of been fun to talk about. These are kind of two indirect applications of our understanding of DNA repair from BRCA and BRCA2. We tried to extend both without a clinical story, because that's something David would have loved to see us put this in the clinic, and actually inspired by Judy Garber, who did the original platinum trials here. And then really something new, which is I want to understand the rearrangement structure, because that's really, in some ways, why is the DNA repair defect selected for? It's selected for to create rearrangements. And the rearrangements, hidden in the rearrangements, are the drivers of these tumors. And we got to hit those as well if we can identify those, if we can. With that, I'll stop. These are my acknowledgements, and happy to take questions.